All right, hi everybody. Welcome to our second lecture. This one is on cellular respiration. Now, let me just preface this by saying this is probably one of the hardest concepts to get in biology. And the reason why is because it's incredibly abstract. There's nothing that we can really compare it to that you're used to on a normal day-to-day life. And so the biochemistry of it can be a little bit abstract. It's a little bit hard to follow. But the way that I kind of like whittle it down, dumb it down, I don't mean to say dumb it down, but like the way that I simplify it is I take just what's important, and that's just tracking the movement of energy through the systems, right? And so what we're looking at is we're looking basically like how does a cell take glucose into its in through its membrane? How does it take in glucose? What does it do with glucose? And then how does it take like the byproducts of the breakdown of glucose when it breaks glucose down? How does it take those byproducts and then convert those into even more energy? And then finally, like how does it do like this energy currency exchange where it takes things that are like $5 bills and $2 bills and turns them all into $1 bills? And so that's the part, like that's kind of like what I want you guys to get out of this is where does the energy come from? It comes from the glucose, right? Where does it get stored in? And then how are those storage molecules, like the cellular batteries, right? How are those used and how do they exchange their energy for ATP at the end? But most importantly, like though, glycolysis is super complex, don't get me wrong. It's got a lot of moving parts, but it's important to study it because glycolysis is the set of primary reactions that are used by every living organism on the planet. Every living organism on the planet. Everything with a cell can do glycolysis, which means it's so primitive. That means it's so ancient. And so that's why it's one of the most, that's why it may seem like it's a little bit advanced for a freshman biology class. You're gonna be like, holy cow, this is like biochemistry. It is biochemistry. When I took my 365 and at ASU, which was biochemistry, we literally like did glycolysis again. So yeah, it's used in a lot of classes. So anyways, let's get started with the glycolysis. But let me give you an overview of cellular respiration to begin with. So let me just orient you to this photo. I know there's a lot of circles and lines going on, right? And I'm gonna pull out my pointer, which is kind of nice. It makes this little red streaky thing. Okay, so in the background over here, right, where you see pyruvate oxidation, Krebs cycle, electron transport chain, notice this is the mitochondria, okay? Notice that primarily the most of what we're talking about, like what we get out of the cell respiration, is going to end up coming from the mitochondria. Glycolysis, we don't really capture that much ATP from glycolysis. We capture some, and we do capture some NADH. And so the ATP is the goal, right? That's what we want. We want to make as many $1 bills as we can to fuel reactions in the cell. But NADH, if there's a step in the breakdown of a molecule where there's enough free energy released that we can capture a $5 bill's worth of energy, we're going to do that. We're going to capture it in a $5 bill. And so that's what NADH is. It's like we're capturing all that free energy in a $5 bill. But eventually we're going to have to exchange that $5 bill for some ones. And so notice that FADH2, one of our other cellular batteries, is going to come out of the Krebs cycle along with some more NADH. And then those are going to go to this electron transport chain where they're exchanged for ATP. And that's where you see all this ATP. So notice all of the arrows eventually lead to ATP coming out of this adenosine triphosphate. But let's start at glycolysis. Okay, so we have a glucose molecule. It's a 6-carbon sugar. We know this is the shorthand method, right? Because all we see are the functional groups attached, right? All the carbons are made up of these points. So you got a line coming here to a point, a line coming here. So this is our first carbon, second carbon, third carbon, fourth carbon, fifth carbon. And then the sixth carbon is labeled as a C because it's sticking off of the structure here and it's got this other stuff bonded to it. And so even though these don't say C next to them, there are 6 carbons here. And then for every carbon in glucose, there's going to be an oxygen and 2 hydrogens, right? That's the carbohydrate or the water that is present with every carbon in a ratio. Okay. Anyways, so what's going to happen is we're going to take glucose and we're going to break it down into a 3-carbon sugar called pyruvate. Now you might say, okay, hold on a second. There's 6 carbons in glucose and there's only 3 carbons in pyruvate. Well, that's because we get 2 pyruvate molecules for every glucose. So I didn't draw a 2x here right, to say that we get 2 of these. But the idea is that glucose gets broken down and oxidized into pyruvate. Something else I want to point out too is that when we talk about reduction and oxidation, like redox reactions, reduction is this idea that when you get electrons, like you gain free energy, like you can do more work, right? Also, one of the things that we noticed was that when things get reduced, they tend to get like a hydrogen attached to them, right? Like an H comes in from somewhere. And I've told you, like, okay, that H represents electrons. When I distinctly remember being a freshman in biology, like this class, and I remember being so confused by that. I was like, how? I mean, because they would always it was almost like my science teacher wouldn't even address the fact that there was a hydrogen being attached, like to NAD. Right. So he wouldn't even address that. He would just be like, "Oh yeah, it's going to gain electrons and then it becomes NADH." And I'm like, "Wait, if it gains electrons, why is it gaining like a hydrogen? Like that doesn't make." And he never addressed it in class, but I want to address it now. And the reason why is because hydrogen acts like an electron carrier. And so when an electron comes in and NADH grabs that electron from the breakdown of glucose, the hydrogen just sort of attaches itself to NAD as sort of a cork on the bottle, if you will. It keeps that electron trapped inside of NAD. So it's like the electron comes in, the hydrogen attaches as a cork to hold the energy in the battery itself. Okay. So, anyways, I digress. But coming back to my central point here is that anytime you see hydrogens bonded directly to a carbon, that molecule has more free energy than a molecule that has oxygen bonded to a carbon. And so really what we're looking at in this entire course when we're talking about the biochemistry of it, we're looking at the oxidative and reductive states of carbon. So for instance, like if you go back and look at like a hydrocarbon tail on a fatty acid, these are the ones that are those long squiggly things sticking off of a phospholipid, like in our phospholipid bilayers, right? They're non-polar. Those tails are entirely made up of carbons bonded to hydrogens, which means they're like, like saturated fats or unsaturated fats. And then I told you guys, hey, upload a nutrition label, because on the nutrition label, it shows you that 1 gram of fat has more than twice as many calories as 1 gram of sugar or 1 gram of protein. And the reason why is because those hydrocarbon tails are all hydrogens bonded to carbon. That carbon is in a highly reduced state, a highly charged up state. Like gasoline, flammable materials usually have these hydrocarbons, carbons bonded to hydrogens. And that's the idea of these like energetic electrons being close to carbon. Now, once you start attaching oxygens to carbon, like look over here on pyruvate, you've got a carbon right here, center section, and that's bonded, double bonded to an oxygen, single bonded to another oxygen. That carbon is in a highly oxidized state, or discharged, if you will. So it's charged up over here where it's got some hydrogens attached to it, and then it's uncharged over here where it's got these oxygens attached. And so in the process of oxidizing glucose into pyruvate, every time something's being oxidized, right, something is also being reduced. And so we have these uncharged or oxidized cellular batteries that then get reduced, right? The transfer of electrons from glucose into these batteries, right? They get reduced, and glucose itself becomes oxidized. And so then you get these charged up, or technically reduced because they're gaining electrons, cellular batteries. So it's the idea of a transfer of energy, right? There's glucose, which has a ton of energy in its bonds. It gets converted into pyruvate, and as it does that, it's being oxidized. So electrons are being taken from it. And those electrons are then being placed into these cellular batteries and charging them up. And then if you remember, NADH is that $5 bill. And so we have to take that to our currency exchange in the electron transport chain later on to be converted into ATP. So anyways, this is glycolysis. This is the idea, the basic concept of it. But let's get a little bit more nitty gritty. Ooh, I almost forgot. All right. So even though we're transferring the energy from glucose into these cellular batteries, some of that energy in that process is being lost to the environment in the form of heat. And so the end state, if we were to take all of the free energy in ATP, NADH, and pyruvate, right, we should be able to add up all of the free energy in those 3 molecules and compare it to the original amount of energy in glucose. And what we'll see is that there's actually, even though we charged up cellular battery, the total amount of free energy has actually decreased, right? Even though we transferred some to other molecules, the total amount of free energy has decreased through glycolysis, which means, right, you take the final state, which is less, subtract it from the initial state, which had more, and you end up with a negative number. And so we lost free energy from the conversion of glucose to pyruvate and charging up these cellular batteries. Even though we lost energy, right, what we're doing is we're coupling that spontaneous reaction, right? Because this is spontaneous, right? This is an exergonic or free energy releasing reaction. We've coupled that reaction to the charging up of cellular batteries. So when glucose is essentially being combusted, right, we're capturing the combustive energy and putting it into cellular batteries. Okay, I digress. Carrying on. So the first question we come to, right, if we go back to those diagrams where we have like the free energy and the energy of activation, right? So every spontaneous reaction, if the reactants, right, those are the things that are going to react. If the reactants are stable in their environment, right, they're not just going to, they will eventually over a long period of time convert themselves into the products, which would have a lower free energy state. So that would be a spontaneous reaction. But if they're just chilling at room temperature, they're not going to just do that like in a minute or in an hour or in a day. It might take them 5 million years, but they'll do it. We don't have time for that nonsense, right? So what we have to do is raise those reactants above their energy of activation by putting a little bit of free energy into the system. That gets the molecules in a state of high free energy, right? And they're a little bit more unstable and shaky. And then we can bring them through the catabolic reactions that release free energy from glucose. So how do we do that? So the first thing we do is glucose is allowed into the cell by just simple diffusion. So consider this, right? You just eat a donut, right? Donuts, tons of glucose, okay? Tons of free energy, tons of sugar rush, right? So anyways, you eat a bunch of, you don't have to eat a bunch of donuts, you just eat one donut. So you eat a donut, right? The sugar from the donut enters into your bloodstream. Your pancreas is a sense organ that says, holy cow, there's a lot of sugar in my bloodstream. This sugar is going to cause some irreversible damage to some of my tissues through oxidation. Sugar can oxidize things. So sugar is being a big problem. It's an inflammatory molecule. So the pancreas says, hold on, I need to regulate this blood sugar. It releases insulin. So insulin acts as a co-transporter to the cells. To allow glucose to flow from the bloodstream into the cell. So insulin is released into the bloodstream. Insulin binds to receptors on the cell membrane, and it opens up a channel or a portal. It's just a gate. It opens up the portcullis. Now, remember that diffusion happens spontaneously. So if there's a higher concentration of glucose in your bloodstream than in your cell, as soon as that portcullis opens, glucose diffuses into the cell. And so it'll do that, right? That's how blood sugar is regulated in the body. So insulin's going to open that door and let glucose come in. As soon as glucose enters the cell, it gets phosphorylated. That means a phosphate group is taken from ATP and placed onto glucose. Now remember, every time that there's a reaction like that, it's facilitated by an enzyme. And the enzyme that does that is hexokinase. So, of course, this is over our heads a little bit, but hexo, meaning six, right? Hexagon, right, is a six-sided thing. Glucose is a six-carbon sugar. Kinase, right? Anything that's kinase is going to be involved in moving a phosphate from ATP onto an enzyme, or vice versa, from an enzyme back to ATP. So kinases are enzymes that deal with the movement of phosphate groups. So it's a six-carbon, a hexokinase, a six-carbon phosphate moving enzyme. So anyways, so glucose gets a phosphate group attached to it, and we'll see a picture of this in a little bit. Okay, that's going to get glucose closer to its energy of activation threshold, but it's not quite there yet. What it does do, though, is it keeps glucose from, it made glucose bigger because we attached a phosphate group to it. And so it keeps it from actually flowing back out the membrane channel that it diffused into. So it kind of traps glucose in the cell. All right. So then what the cell does is so the glucose has a phosphate group on it, right? So that makes it glucose 6-phosphate. But the problem is there's nowhere else to put a phosphate group on glucose, right? So what does the cell do? It's going to rearrange the glucose structure so that now it's got another arm and it's actually fructose. So remember, glucose and fructose have the same chemical formula. And so all we have to do is rearrange their structures to allow for another attachment of a phosphate group. And so that's what happens. So phosphoglucose isomerase. So that's the enzyme that facilitates the rearrangement of glucose into fructose. Okay. So phosphoglucose isomerase gets its name from, if you have a glucose and it's phosphorylated and you need to make an isomer of it, use this enzyme. That's basically all that word means. So it's a big word, but it's not just a random naming of a molecule. It's actually like functional too. So anyways, it rearranges it. So now it's fructose 6-phosphate. Okay, so the phosphate is still bound to the 6 carbon. Now it allows for room for another phosphate to be added. So there's another phosphofructokinase. There we go, right? That should say phosphorylated. There should be an R there. Phosphofructose kinase takes a phosphate group from ATP and it sticks it onto glucose again. Or, well, it's not glucose anymore. It's fructose 6-phosphate. So then it becomes fructose 1,6-bisphosphate. Now we have reached our energy of activation. So we've added free energy to our glucose molecule, brought it up, its energy of activation a little bit higher, and now we're ready for it to be broken down completely. So anyways, like I said, fructose 1,6-bisphosphate is that molecule that has 2 phosphates attached. Okay, good news. So anyways, let's take a look at each one of these steps as we're moving forward. So this is where we're raising glucose above its energy of activation, right? So we have glucose here. Hexokinase is going to take a phosphate group from ATP and attach it onto glucose. That makes glucose 6-phosphate. Notice there's this yellow P right here attached to that O. So in order to attach it, remember, in order to attach this, we have to dehydrate this reaction. So this hydrogen pops off, and OH groups pops off, this OH group pops off a hydrogen from the phosphate. That makes water. So what you don't see is a water molecule leaving, and then this phosphate group attaching itself to that carbon, to that 6 carbon. And you might say, well, look, there's an O here and there's an O here. Right, that O right there, that came from the phosphate. And this OH right here left in the water molecule. So I know that's a lot of chemistry, not stuff you have to regurgitate on an exam, just sort of like a functional explanation of what's happening here. Okay, so glucose has nowhere else to put a phosphate group on it. And so the phosphoglucose isomerase changes where this oxygen is bound to. It changes it, moves that bond down to the 2 carbon. So now that oxygen is bonded to the 2 carbon, and the 1 carbon is the one sticking up here. And so we still have 6 carbons, 1, 2, 3, 4, 5, 6. We still have 6 carbons, right? We have a phosphate attached to the 6 carbon, and it's a fructose molecule. So fructose 6-phosphate. Now we've created space for another ATP to drop its energy off onto fructose 6-phosphate. That's facilitated by phosphofructokinase. Remember, kinases move phosphate groups from molecule to molecule. And so now we have fructose 1,6-bisphosphate, which has 2 phosphates, one on the 1 carbon, one on the 6 carbon. Bis means 2, right? Like bi. Okay, so that's where you get that. Now, the cell at this point, right, this molecule has a ton of free energy, right? It is unstable. It's ready to be broken down. So the cell comes along and it cuts it with a pair of scissors called aldolase. So aldolase is an enzyme that makes an aldehyde. So here's our glyceraldehyde. Now, going back, I know we're just throwing terminology at you and you're like, what the heck do I have to know? I totally get it, right? So follow the note sheet. Don't forget, just follow the note sheet, right? The stuff you need to know is on the note sheet. Definitely try to understand what I'm saying. Try to follow like try to follow the concepts as we move forward. You don't have to memorize these names. I'm going to explain where the names come from, but you don't have to memorize that. Okay. So anyways, so aldolase makes an aldehyde whenever you have a 3-carbon sugar, right? Because we had a 6-carbon sugar here, we cut it in half. So now we have 2 3-carbon sugars, right? They're not going to look exactly the same because they're mirror images of each other, but we're going to have to rearrange the dihydroxyacetone phosphate. We're going to have to rearrange that into a glyceraldehyde 3-phosphate in a second, but we're not doing that yet. Okay, so aldolase cuts the fructose 1,6-bisphosphate in half. Aldolase creates these 2 molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, notice, right, 1, 2, 3 carbons here, 1, 2, 3 carbons here. Okay, we're going to take an isomerase, which, if you think about it, is an enzyme that helps to isomerize or make an isomer out of a molecule. So that isomerase is going to take dihydroxyacetone phosphate, DHAP, and it's going to isomerize it into glyceraldehyde 3-phosphate. And we'll see that in a second. So let's talk about the free energy steps that we've taken so far, right? So here's our diagram, right? So we've got free energy up the y-axis, and then how the reaction is proceeding forward on the x-axis. So we've got glucose, right? We add 2 ATP to glucose, right? That brings it above its energy of activation. Without an enzyme, it wouldn't break down. But with an enzyme, right, the energy of activation has been lowered, right, with the enzyme. Without the enzyme, it's way higher. You'd have to add way more energy to glucose to break it down. But with the enzyme, we only have to add 2 phosphates, and then we get a release into pyruvate. So we're going to break down this molecule. We're up here at the top. We're going to break it down and turn it into pyruvate. Remember, we had 2 molecules of glyceraldehyde 3-phosphate, right? And so we're going to end up with 2 molecules of pyruvate, which are 3-carbon molecules as well. So we're just going to oxidize glyceraldehyde 3-phosphate into pyruvate, and that oxidation reaction is going to help us reduce some other molecules. So glucose was phosphorylated twice, right? We rearranged it to make space for the phosphate group. This added free energy to the system. Glucose was then split in 2. That's making the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. One of them was isomerized, right? So we isomerize it into G3P. And then the exergonic portion of glycolysis will begin with 2 G3P molecules. And that's where we are here, right? 2 glyceraldehyde 3-phosphates. Now, in a big step, right, watch, this is a big drop in free energy here, a big oxidation, because we're going to take 2 of these glyceraldehyde 3-phosphates, and we're going to charge up 1 NADH from each one of them, okay? So 2 NAD pluses come in, right? 1 for each one of these glyceraldehyde 3-phosphates. 2 electrons come in, right? Those are carried by the hydrogens. Remember, that's why the hydrogens get attached. So we rip off those electrons, which means we're oxidizing G3P into 1,3-bisphosphoglycerate. Now, that drop in free energy was so great that we can actually take 2 more inorganic phosphates and attach them to our molecule. So we oxidize this molecule into this molecule and also attach the phosphate group to it. Okay. All right. Now we can start to capture these phosphates back in the forms of ATP. So remember, we have 2 1,3-bisphosphoglycerate molecules, right? There's going to be an enzyme that facilitates the gathering of the phosphates and putting them back on the ATPs. Okay. So that happens to each molecule once, which yields 2. Then we have 2 of these 3-phosphoglycerate molecules. There's going to be an enzyme that helps to rearrange it. So it rearranges that phosphate group and puts it on the second carbon. See how the P changes to be on the second carbon? Then we remove some water from the process. And then we take that last phosphate from those 2 phosphoenolpyruvates and we place them back onto ADP and make 2 more ATP from this process. And that yields pyruvate. And so what I really want you to know, right, is yeah, you saw way more details than what I want you to know. But just being able to see, like, okay, we got to add energy to glucose to make it unstable, right? That cost us some ATP. And then we get those ATPs back, right? We get it in the second step here, right? It cost us 2 ATP to break down glucose. We caught them back here. So those 2 cancel out. So our net gain of energy from glycolysis is 2 NADHs and 2 ATPs, right? So energy gains, right? We gained 2 NADHs and 4 ATPs, but we had to spend 2 ATPs up front in order to even get those. So the net gains are 2 NADHs and 2 ATPs. Okay, and so next, we've got 2 pyruvate molecules, highly oxidized, but they do have, look, they've got this carbon right here that's bonded to 3 hydrogens. This carbon right here is highly reduced, right? Think that's a methyl group, right? It's a bunch of these that make up the hydrocarbon tail of a fatty acid. They give it all of that 9 calories per gram, right? So a lot of free energy here. And then look up at the top of the molecule, though. There's carbons that are bonded exclusively to oxygen. So one side of this molecule is highly oxidized, and one side of it is reduced. And so we need to take the reduced side of this molecule and go get some more free energy out of it, right? And so that's the byproduct that's going to be used in the Krebs cycle. So let's take a look at this. So once we have pyruvate, it kind of enters into a fork in the road, right? There's a question that the cell has to ask. And that question is there oxygen present? Okay, so this has to do with aerobic respiration and anaerobic respiration. So your cells are all doing glycolysis no matter what, right? But let's say you're like exercising, right? Maybe you're jogging down the street. Maybe your cardio is good enough that the amount of oxygen you're absorbing from the air is sufficient to power your mitochondria. But if it's not, right? If it's not, right? It's going to have to go do something else because the mitochondria only works if there's oxygen present. And so if there's oxygen present, right, then it can go into aerobic respiration, right? So we can take that pyruvate into the Krebs cycle, into the mitochondria, and we can exchange it for way more energy than we would if we just had to do glycolysis. So aerobic respiration is huge. It yields so much free energy, and you'll see why in just a second, okay? If it doesn't, if there's no oxygen, let's say that you're doing your jog and your cardio sucks and you're starting to gas and you're like breathing hard, right? Then you don't have oxygen to power your mitochondria. And so you're going to have to keep the pyruvate in the cytoplasm of your cell and it will get fermented. And that fermentation actually removes some of the electrons from NADH. So it actually takes some of that free energy back in order to make more NAD+, because NAD+ is an oxidized, uncharged battery. And so the cell recognizes that and is like, holy crap, I need to bring in more glucose. And so that stimulates the cell to bring in more glucose. But let's focus on the first one right now. So is there oxygen present in the cell? If the answer is yes, then we're going to take this sucker and bring it into the mitochondria. And so what you see over here, right, you see the cytoplasm, right? So that's where the pyruvate is being generated, okay? Mitochondria is on the inside of this yellow membrane, this plasma membrane that separates the cytoplasm from the mitochondria. So first of all, when pyruvate enters the mitochondria, carbon dioxide is going to get removed from that pyruvate, okay? That process of removing carbon dioxide actually steals an electron from the remaining molecule. So that means that it's oxidizing the reaction, right? So carbon dioxide oxidizes pyruvate a little bit when it pops off, and that oxidation reaction allows NAD+ to collect an electron from that reaction and become NADH. So count that, right? Like, let's look at the energy gains, right? So from glycolysis, the net gains we had was 2 $5 bills and NADHs and 2 $1 bills. So the net energy gain in currency from glycolysis is 12, right? 2 NADHs, that's 10, and then 2 ATPs, that's 12. Okay, so now let's see what the net energy gains are of the Krebs cycle. So remember, right? We have 2 pyruvate molecules. So we're actually getting 2 NADHs from this whole process right here, just from pyruvate getting in the mitochondria. Okay, so carbon dioxide pops off. Another thing I want you to keep track of, right, is notice the number of carbons from glucose, right? We started with 6, right? Now we have 2 3-carbon molecules in pyruvate. Notice the number of carbon dioxides generated from the oxidation of glucose. Watch. We're getting 2 carbon dioxides here, right? Because we ended up with 2 pyruvates at the end of glycolysis. So now we're getting 2 carbon dioxides that pop off. So that's 6 total carbons minus 2 here. Now we have 4 carbons left. So those 4 carbons are distributed between 2 pyruvate molecules. They're no longer pyruvate, right? Because they've lost that carbon. They're no longer pyruvate. They've lost that carbon. Sorry, my cat's climbing on my computer. See, look. So anyways, they've lost those 2 carbons. So they're not actually pyruvating anymore. They are an acetyl group. Dude, your tail is like in front of the camera, kitty. Don't smack me. Okay. Where was I? Sorry, the cat's like totally rubbing on my like microphone here. Just lay down, kitty. So anyways, when they've lost that carbon dioxide, right? Those 2 pyruvates have lost 2 carbon dioxides, one each respectively. Now they're down to an acetyl group. Now there's a transport molecule that comes in from the middle of the Krebs cycle that basically feeds the Krebs cycle, these like oxidized pyruvate molecules. So this coenzyme A here comes and it binds to the 2 carbons that are left over after this 1 carbon dioxide left, right? And so we've got these 2 carbons here, right? One of them is gone in carbon dioxide. And those 2 carbon molecules, we call that an acetyl group. So this is acetyl coenzyme A. Acetyl coenzyme A. And that's what that's what gets transported into the mitochondria. Okay, so coenzyme A is a transport molecule. What are you? Oh my gosh, cat, what are you doing? It's a transport molecule binding to the remaining acetyl group. And here, you're gonna have to get down, kitty. And then that's transported into the mitochondria. Okay, so don't freak out. The graphics changed a little bit here. So now we're in the Krebs cycle. So this is that final stage of where we can take the carbons from glucose and finish breaking them down into carbon dioxide. So in this peach box up here, you'll see what we've already done, right? So we've taken this 3-carbon pyruvate, ripped one of the carbon dioxides off, charged up an NADH from that process. Coenzyme A bound to the remaining 2 carbons, and it's going to transport those 2 carbons into the Krebs cycle, right? Okay. So where was I? Okay. So acetyl coenzyme A is going to drop the remaining 2 carbons off. Now those 2 carbons, right, that acetyl group is going to bind to a molecule called oxaloacetate. And oxaloacetate is just a 4-carbon molecule that's finishing the Krebs cycle, so it's already gone through the Krebs cycle. As soon as this 2-carbon molecule bonds to this 4-carbon molecule, we end up with a 6-carbon molecule called citrate. And so these 2 carbons get dropped off this 4-carbon molecule. When they get dropped off, this shuttle of coenzyme A is going to transport itself all the way back out towards the cell membrane or the mitochondrial membrane. It's going to grab the next pyruvate that enters and gets oxidized and grab the next acetyl group and bring it in. And so this shuttle coenzyme A is just doing a loop, right? It has like a shuttle route, if you will, right? It picks up, it drops off, it picks up, it drops off, it picks up, it drops off. When it drops off, it's adding 2 carbons to oxaloacetate, making it citrate. Okay, so now check this out, right? As citrate goes through the Krebs cycle, there's a bunch of enzymes here. I didn't picture them, right? You can go look it up if you like. I'm sure there's plenty of YouTube videos because you have to memorize the Krebs cycle and you have to memorize glycolysis, all the steps, all the molecules, if you're a pre-med student. So another reason why you learn this in freshman biology, right, is there's a ton of enzymes that are working here, okay? And what those enzymes are working to do is they're working to break down citrate into oxaloacetate. They're working to oxidize citrate into oxaloacetate. In order to do that, 2 carbon dioxides will have to be removed, right? These 2 carbon dioxides that enter, or these 2 carbons that entered from the acetyl group will eventually become oxidized and become carbon dioxide. Now that oxidation process of releasing those carbon dioxides is enough of an oxidation that we can actually charge up 3 cellular batteries here. 2 are going to be NADH, 1 is going to be ATP, and the other one is going to be FADH2, and then we'll get 1 more NADH up here. So when I say 3 different types of cellular batteries, right? Now remember, this is just from one, right? This image in this graphic is just from 1 pyruvate. Right? Remember, we have 2 pyruvates doing this for every molecule of glucose. So let's back up and count our carbons here, right? Pyruvate, right? We have 2 pyruvates that exited glycolysis. We're going to lose 1 carbon from each of them. So now we have 4 carbons, right? So 2 of the groups that are entering the citric acid cycle or the Krebs cycle. So 2 of these carbons from each one. So that's 4 total. And then in this cycle here, we're just following what happens to one of them. So you have to double all of these numbers if you're accounting for all the carbons from glucose. So there's actually going to be 4 carbon dioxides lost. That accounts for the rest of the carbons from glucose. There's actually going to be 4 NADHs charged up here, 2 FADH2s here, and 2 NADHs here at that last step. And so if we add those all together, right, let's do that real quick. So for the Krebs cycle, we have 2 NADHs here, right? 4 here, that's 6, 7, 8. So we actually get 8 NADH, 8 $5 bills once you enter the mitochondria. Go back, think about what we got from glycolysis. We got 2 $5 bills from glycolysis for 1 molecule of glucose. Once it gets to the mitochondria, you get 8 $5 bills. That's a huge difference. That's like 3 times as many. And that's just the $5 bills. Look, we also get 2 ATP from this and 2 FADH2s. FADH2s are worth $2 bills, so that's $4 there, $2 there, $40 there. If you take the 8 times 5, that's 40, right? 4, 5, 6. So we're essentially getting $46 worth of energy from the mitochondria, where we're only getting $12 worth of energy from glycolysis. So that's why it's like aerobic respiration is so sustainable as long as you have oxygen, right? So we already talked through these steps. Okay. So let's go back to our energy dilemma, okay? The problem is in the cell, Most reactions only cost $1, right? They only cost 1 ATP. And you do not get change back for those reactions. The change is lost in the form of entropic energy or whatever, electromagnetic radiation, heat, right? Unable to do any work, right? That change just gets lost into the universe. So a $5 bill, if it only costs $1 to do a reaction or less, right? To use a $5 bill to power that would be a waste of energy, a colossal waste. Even using a $2 bill would still be a waste of energy. And so we have to do this currency exchange with NADH and FADH2 and try to get ATP back for that. And that's where the electron transport chain comes in. The electron transport chain is literally a currency exchange center in the mitochondria to get more ATP. Now, it's incredibly complex, right? I'm not going to lie. Look, this is your mitochondria, right? Or an artistic depiction of one, right? And remember that it had two membranes, right? The pictured on the inner membrane here, usually when you see the mitochondria drawn, it has a bunch of folds. It's got a bunch of like squiggles on this inner membrane, right? And then the outer membrane is usually a oval shape like that. Okay. All right, where the magic happens for the electron transport chain is in the middle of the mitochondria, right? That's where the Krebs cycle is taking place. So notice the Krebs cycle is happening over here. And the Krebs cycle, if you think about it, is producing tons of NADHs, right? And tons of FADH2s. So let's talk about the subunits of the electron transport chain. There are 4 subunits. And they have names, but I don't care if you know them. Okay, we'll just call them subunit 1, 2, 3, and 4. Okay. In this top chain up here, there's only 1, 3, and 4. In this bottom chain down here, you can see 2, 3, and 4. And so the reason why they did this is they're showing you where FADH2 drops its off at, like what subunit FADH2 drops its electrons off at, and then what subunit NADH drops its electrons off at. Okay. So what you can do is you can think about these subunits that are embedded into this membrane as being electrical pumps. And so when the electrons get dropped off on them from NADH and FADH2, when the electrons get dropped off, the electrons power these pumps. And what these pumps do is these pumps pump protons. See these 2H pluses, 2H pluses, 2H pluses. These pumps, when they're operating with electron energy, just like an electric circuit, right? Drop electrons off, they flow through a pump, the pump works. As the electrons flow down this electron transport chain, right? It operates these pumps that pump these protons into the intermembrane space. And so remember, anytime that you have just a hydrogen proton, excess hydrogen protons, right, in a solution, you're going to have an acid. And so that's going to be a low pH, high concentration of hydrogen protons, very low pH, very acidic. And so basically what these electrons are doing from NADH and FADH2 is they're making the intermembrane space very acidic with a whole bunch of hydrogen protons, right? And those hydrogen protons are just being pumped into that space over and over and over again as these pumps are working. These pumps are powered by the electrons from FADH2 and NADH. And so NADH drops, this is literally why NADH is worth a $5 bill, because NADH drops its electrons off on pump 1, on subunit 1, and it pumps 2 protons. It then takes that electron and it gets transferred, right? It flows down the wire to subunit 2. And that subunit 2, right? It pumps 2 protons. Subunit 3 pumps 2 protons. Subunit 4 pumps 2 protons. And so the earlier that you drop off electrons on these subunits, the more protons get pumped into the intermembrane space. And those protons are what power the conversion of those protons are what power ATP synthase, which is what generates ATP, right? So that's how we convert it. So you can think about it like, oh, electrons get dropped off on the subunits by NADH and FADH2, right? So that's our $5 bill dropping off its money, its currency, right? So $5 bill drops off its currency, FADH2, a $2 bill drops off its currency. The currency is electrons. They power the pumps. The pumps pump protons into the intramembrane space, reducing the pH, increasing the acidity of it, right? The electrons flow through those subunits. They're pumping the protons. And then once those electrons get to subunit 4, this is where it gets important, right? This is where oxygen comes in. So those those electrons that are flowing down the wire of the electron transport chain, they're eventually going to find 2 protons and half of an oxygen gas molecule. So you can think like, O2 is what you breathe in. It gets split, so now you have 2 oxygens, 2 protons, 2 electrons, right? That makes a water molecule. So the oxygen you're breathing in, your mitochondria is using that to collect, to attract the electrons down this wire, because oxygen's electronegative, right? So those electrons are attracted to the oxygen. As soon as they get there, right, that oxygen that's sitting there grabs those electrons, 2 protons pop on, and boom, you got water. And so if you think about it, that's a very clean energy reaction, right? You're just taking oxygen you breathe and turning it into water. So that's pretty cool. So that's how that works. Now, so that's what happens to the electrons. The electrons end up back on water. Now remember, all of these protons are building up in this intramembrane space, right? So this compartmentalized reaction is allowing us to increase the concentration of hydrogen protons, decreasing the pH, making it incredibly acidic out here. But consider this, right? I've always told you this, molecules are magnets, right? So if you have a whole bunch of positively charged ions all compressed together inside of this little space, oh my gosh, all they want to do is diffuse and get away from each other, right? They're all of the same charge. And so what ATP synthase does is it allows a channel, like a water wheel, right? It allows a channel for these protons to flow back into the middle of the mitochondria. And as they flow into the middle of the mitochondria, right, through this water wheel, that water wheel is literally spinning. It's ratcheting, just like a turnstile, like at a subway, right? It's ratcheting every time that something flows through it and spinning it. The rotational energy of this subunit is what creates the energy and power to smash a phosphate group onto an adenosine diphosphate. Bam! And it makes ATP. And so that's how you get ATP, is it's this flowing of protons back into the matrix. And then those protons get picked up by these pumps that are being powered by NADH and FADH2. And those protons get pumped back out into where they were not happy. And they're like, no, not again. I'm going to flow back through this ATP synthase. And so they're getting pumped in, or sorry, they're getting pumped out, flowing in, diffusing in, powering a water wheel that basically converts that rotational torque energy into ATP. And so that's how you convert NADH and FADH2 into ATP. All right, so a little summary here, right? So pyruvate goes to the mitochondria, right? 2 pyruvates for every glucose. It goes to the mitochondria and enters the Krebs cycle. Eventually, it gets oxidized into carbon dioxide. So think about that, right? You've got glucose. The electrons from glucose are taken away. Cellular batteries capture them, right? Glucose is oxidized into pyruvate. Pyruvate's oxidized into carbon dioxide. So glucose eventually becomes carbon dioxide, CO2. The electrons are captured by the batteries, taken to the electron transport chain, right, and then converted into ATP. And so coupled with the oxidation of pyruvate in the Krebs cycle, right, NAD plus, FAD, ADP, plus an inorganic phosphate are all charged up. So that's all I was saying there, is that the cellular batteries are getting charged up from the electrons from glucose. They're getting reduced by those electrons. Okay. NADH, FADH2, they do their electron exchange, right? They drop them off on the subunits. Subunits pump protons, right, into the inner membrane space. Electrons flow down the subunits, powering the pumps, right? As soon as they get to subunit 4, oxygen, protons, electrons combine and form water. So the protons build up in the inner membrane space, right? They're all positively charged ions. They're repelling each other so hard they want to get away. ATP synthase acts like a water wheel or a turnstile. The torque of it rotating is enough to transfer that rotational energy into ADP and an inorganic phosphate, thus making ATP. It's pretty cool. It's pretty dang clean, too. Glucose becomes carbon dioxide. The electrons become water. No toxins, no pollution, no nothing. It's pretty sweet. Okay, so anyways, let's go back to pyruvate's dilemma, right? So we asked, is there oxygen present? Yes, that's in the mitochondria. That's what powers everything, is that oxygen sitting on subunit 4. Okay, so we already covered aerobic respiration. Let's look at anaerobic respiration, right? What happens if there's no oxygen present, okay? All right, so let's take a look in prokaryotes. Prokaryotes and actually yeast. Yeast is a weird one. Yeast is a single-celled fungus. So it acts kind of like a protist in that it does the same sort of fermentation that a protist does, except its byproduct is ethanol, right? Yeast, beer, ethanol. Okay, fermentation, sound familiar? Wine, hey, all right. So anyways, glycolysis is gonna produce a bunch of pyruvate, right? And so this is all in the cytoplasm, right? So pyruvate gets this is in prokaryotes only. There's 2 different pathways. One is in prokaryotes, one's in eukaryotes. So prokaryotes and yeast, right? This pyruvate that's created from glycolysis, it says, oh no, there's no oxygen present. Okay, quick, let's make some more NAD plus so I can bring in more glucose so I can actually get some net ATP to do some work around here without oxygen. So that pyruvate's gonna lose its carbon dioxide. Guess what? That's the bubbles in beer, right? So that's the bubbles in bread too, like when you're making bread and it's rising. That's the yeast inside of the bread. The yeast is taking in the glucose from the starch, right? So starch is a bunch of glucose bonded together. Yeast is taking the glucose from the starch, okay? And then it's taking the glucose, going through glycolysis. There's not enough oxygen present in the middle of the bread, so the yeast basically says, all right, screw it. I'm going to release carbon dioxide. I'll make acetaldehyde, okay? And then I'm going to take that NADH, that $5 bill that I cannot use, right? And I'm just going to take its electrons and I'm going to put it on this acetaldehyde and make ethanol. So consider this, right? You're taking a high energy molecule, NADH, right, that you've already captured from glycolysis, and you're taking its electrons and you're putting it back into the original molecule. And so what that's doing is it's creating a bunch of dead batteries. It's also taking that energy and putting it into this ethanol, right? My kid's crying. Pause. Okay, I'm back. So anyways, you have this process, right, where we're capturing free energy from glycolysis, right? ATP is great. We can do a lot of reactions with ATP. But because there's no oxygen, we can't take pyruvate to the mitochondria and get those 40, what do we do, 44, 46, right, dollars from that process. We're only getting $12 from glycolysis, right? And 10 of those dollars we can't use. So we're only getting like $2, right, from this whole process. So glycolysis is driving forward. We can't go to the mitochondria. We lose our carbon dioxide, right? Poop, pop that off. We then now have this acetaldehyde is able to capture the electrons from NADH. And so NADH gives its free energy to acetaldehyde and makes ethanol. This is why ethanol is flammable. Look at it. See that central carbon? Look how many hydrogens are bonded to it. Look how many carbons are bonded to this one, right? There's just that OH group, so it's slightly oxidized, but this molecule is highly reduced. This molecule has a lot of free energy. Think Molotov cocktail, right? So fermentation is creating flammable materials, but what it's doing, the whole reason it's going through all this process, the whole reason it's bothering to do this is to bring in this signal, to create more of this signal for more glucose. When the cell has high levels of NAD+, it's like, dude, open up the channel, bring in glucose, quick, we need to drive glycolysis forward. And so it kind of tricks itself, if you will. It's like it biohacks itself to bring more glucose in, to go through glycolysis, to get those $2, right? We need those $2 bills. Or they're not two $1 bills. We need those dollar bills, right? We need those dollar bills for our reaction. So that's the whole purpose with anaerobic respiration, right? We need something to capture the electron from NADH because we can't use NADH. So we need something to take the electron. That ends up becoming ethanol that recycles our NAD plus receptor that signals to the cell to bring in more glucose to drive glycolysis forward. Right. And so you get this cycle, boom, boom, boom. And the whole byproduct is ATP, right? Just trying to get more ATP. All right, eukaryotes, slightly different, actually more simple, if you will. We don't actually lose any carbon dioxide in our fermentation, right? But we still sacrifice our NADH, right? We recycle it back onto our pyruvate. So even though we like create pyruvate from glycolysis, this NADH gives its electrons back to pyruvate and creates lactic acid. And so pyruvate is reduced, right? Because we're putting electrons onto pyruvate and creating lactic acid. And then the NAD plus signals the cell to bring more glucose in. Same thing as the other one, just 2 steps instead of 3. Okay. A meager net of 2 ATP are gained from this process with no oxygen. And so you can't really sustain yourself in anaerobic respiration. So a good example of this, right? Lactic acid is what causes that muscle burn when you're sprinting or doing something that you can't sustain, a muscular activity that can't be sustained for a long period of time. So bench pressing, lifting heavy weights, sprinting, that sort of, maybe running upstairs, that sort of stuff produces that burn, that lactic acid burn, because you're not able to get enough oxygen into your bloodstream to fuel the mitochondria and the muscle cells. And so they're switching into fermentation when that happens. And so that's just kind of fun. So that's the end of this lecture. I hope you guys enjoyed it. I'll see you later.